My name is Priyanka Thirumurti. I work at the University of Buffalo at the Jacobs School of Medicine and Biomedical Sciences. I'm a master's student in the Neuroscience department, and my project is on behavioral analysis of neurodegenerative and neuroprotective effects of two different mutations in the animal model Drosophila Melanogaster. The levy mutation is responsible for neurodegenerative effects, and the suppressor of the levy mutation is responsible for neuroprotective effects. Neurodegeneration is neuronal cell death induced by molecular events involving protein products and pathways within and between neurons involving protein aggregation that can lead to production of neurofibrillary tangles as such or other protein related compounds. In the fruit fly brain, the neuronal cell death is expressed in different regions of the optic lobes of the fruit fly brain. Specifically, my research focuses on two mutations in the fruit fly, one of which causes neurodegeneration, as I specified previously, the levy, and the other which protects it from occurring, the suppressor of levy. I conducted different behavioral assays to understand the effects and the extent and the characteristics of the mutations. I analyzed fly behavior in relation to these types of effects in these two mutations by means of several different assays that differ in relative environmental sensitivity, including space, temperature, and nutrient exposure. For example, the Locomotor Assay utilizes a plastic chamber with a small circular opening at the top where one fly at a time is aspirated into the chamber, and a metal spatula is used to nudge the fly 10 times to see how many times out of 10 the fly will either fly, walk, jump, or display no movement. In my experiments, 100 flies for 5 different strains were used for 500 different flies and 5,000 different data points. In the climbing assay, which is also referred to as a bang sensitivity assay, because the vial with the flies in them is tapped slightly on the tabletop to induce climbing movement, a camera records how many flies are above the 8 centimeter line of each vial every 10 seconds. In my experiments, I tested 250 flies and repeated each set of 10 experiments with 25 flies each, recording data every 10 seconds for 2 minutes, 3 times per experiment for a total of 360 data points. In the lifespan assay, I studied how long it took each genotype to die, transferring the flies exposed to fly food every 2 days and recording the number of dead flies every 24 hours. In the temperature-sensitive paralysis assay, a camera recorded vials with 10 flies of each genotype placed in racks submerged under a 38 degrees Celsius water bath in a tank with a thermometer to indicate changes in temperature as temperature rose in the water tank while it was being heated by switching a knob to calibrate the temperature setting. A total of 5 experiments were completed with 50 flies per experiment and 5 paralysis curves plotted per experiment for each genotype. A paralysis curve shows how long it takes for a specific mutant or control genotype to paralyze and takes the shape of half of a parabola with varying widths on the basis of neurodegenerative or neuroprotective effects. These different behavioral experiments showed high levels of statistical significance in demonstrating the neurodegenerative and neuroprotective effects of the mutations, especially with the locomotor assays and the temperature-sensitive paralysis assays with a p-value of less than 0.0001. I hope my research can be extended into the field of therapeutic interventions of related mitochondrial neurodegenerative disorders by recognizing that these mutations, the levy and the suppressor of levy, which were identified in the context of mitochondrial encephalopathies induce and suppress neurodegeneration, respectively.